Hey, welcome to Pharmacovigilance A to Z, where you learn pharmacovigilance from scratch. If you are new to this channel, please don't forget to subscribe so that you don't miss any of my videos. In my last video, I talked about the ICH, Good Clinical Practice Guidelines. We cannot talk about drug discovery and development without talking about the ICH, Good Clinical Practice Guidelines, because at a certain stage in the drug discovery and development process, human subjects are involved in clinical trials, phase 1, phase 2, phase 3. These good clinical practice guidelines are internationally accepted ethical and scientific quality standards for designing, conducting, recording, and reporting trials that involve human participants. These guidelines ensure that the rights, safety, and well-being of trial participants are protected and that the data generated is credible and accurate. That said, for today, I am going to talk about the drug discovery and development process. On the slide, from left to right, pre-discovery, basic research and screening. 10,000 compounds are screened, 3 to 6 years for the process. Then we move to clinical trials, phase 1, phase 2, phase 3. For phase 1 clinical trials, number of volunteers are 20 to 100. For phase 2 clinical trials, number of volunteers are ranging from 100 to 500. For phase 3 clinical trials, number of volunteers are ranging from 1,000 to 5,000. Years required for clinical trials between 6 to 7 years. Then we move to the next stage, which is feeder review, manufacturing. 1 feeder approved medicine. Then we move to phase 4, which is ongoing research and monitoring. Years required for between feeder review and manufacturing between 0.5 to 2 years. On our next slide is a further breakdown of the drug discovery and development process. From left to right, pre-discovery, basic research and screening. 1. In vitro studies, which is lab testing. 2. In vivo studies, which is animal testing. Then we have clinical trials. 3. Human studies. Clinical trials are divided into phase 1, phase 2, phase 3. Then we have 4. NDA, which is new drug application that is filed to fill up for review. And we have 1 approved medicine. On my next slide is a further breakdown of the drug development process. In this case, is in vitro drug development studies. 1. Target identification and validation. Target is a protein, enzyme, receptor involved in the disease process is identified and validated. 2. Compound screening. By screening, we identify compounds or biologics having activity against the target. These compounds are called hits. Number 3, hit to lead optimization. Hits are refined to improve potency, selectivity, and drug-like properties. Hits are now called lead. 4. Mechanism of action studies. Determine how lead compounds interact with the target and modulate its activity. By doing so, we confirm the drug mechanism of action and target engagement. 5. Cell-based assays. Cell-based assays evaluate the compound's biological activity in cell cultures. By doing so, we confirm the compound's efficacy and cytotoxicity in cellular models. 6. ADMET profiling. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. ADMET properties have been assessed in vitro. By doing so, we eliminate compounds with poor ADMET profiles. 7. Disease modeling. The compounds are tested in vitro disease models. By doing so, we evaluate efficacy and specificity. 8. Toxicology studies. We identify potential toxic effects of the compound. By doing so, we identify toxic doses and refining therapeutic windows. Therapeutic windows refers to the range of drug dosages that are effective without being toxic. 9. Biomarker identification. We identify biomarkers to monitor drug activity and efficacy in future preclinical and clinical studies. 10. Preclinical candidate selection. We choose the most promising compound or compounds for further development. This compound is moved for testing in animals. This is called in vivo studies that I will discuss in my next video. Criteria for preclinical candidate drug selection are high efficacy and potency, favorable ADMET profile, low toxicity, and high specificity. So on my next slide is just a detail of in vitro studies that I just explained. So if you wish to further understand in details, for example, you can go through. Here I mentioned some of the methods used in target identification that include gene expression studies, protein-protein interaction assays, high throughput screening to identify potential druggable target. Concerning compound screening, methods used include high throughput screening of large-scale compound libraries, VITRO screening using computational models, assays to measure binding affinity, enzymatic activity or receptor activation, hit-to-lead optimization, methods used, structure-activity relationship studies, medical chemistry optimization, biochemical assays to evaluate improved efficacy. Concerning mechanism of action studies, methods used are enzymatic inhibition or activation studies, receptor binding assays, and cell-based assays for pathway analysis. As concerning cell-based assays, methods used are testing in disease-relevant cell lines, e.g. cancer cells, neurons, immune cells, cell viability, proliferation or apoptosis assays, measuring functional biomarkers, e.g. cytokine production and gene expression. For ADMET profiling methods, we have metabolism, enzyme stability test with liver microsomes or hepatocytes. We have toxicity, cytotoxic assay, genotoxicity test, and hepatotoxicity evaluations. For absorption, we have CaCO2 permeability assays or PAMPA, which is parallel artificial membrane permeability assay. For drug-drug interactions, we have cytochrome P450 inhibition or induction studies. As concerning disease modeling, methods used, we have 3D cell culture, spheroids or organoids mimicking disease conditions, co-culture system, e.g. tumor and immune cells, use of patient-derived cells or induced pluripotent stem cells. For toxicology studies, methods used acute toxicity test, e.g. LD50 equivalent in cell models, long-term exposure studies in cell cultures, evaluating off-target effects. For biomarker identification, methods used, proteomic and transcriptomic analysis, ELISA, Western blot, or PCR for specific markers. Then, preclinical candidate selection, criteria: high efficacy, potency, favorable ADMET profile, low toxicity, and high specificity, as I said earlier. So, the advantages of in vitro studies. 1. Cost effective. In vitro studies are less expensive than in vivo studies, animal or human trials, due to smaller scale setups. 2. Ethical. Reduces the need for animal testing at the early stages of drug development. 3. Control. Provide a highly controlled environment to isolate variables and study specific mechanisms. 4. Speed. Enables faster screening and analysis compared to more complex in vivo systems. Then now we move to the disadvantages of in vivo studies. Lack of system complexity. That is, cannot fully replicate the complex interactions and systemic effects of drugs in a living organism. 2. Translatability. Results from in vitro models may not always predict outcomes in vivo due to differences in metabolism, immune response, and other factors. 3. Limited representation. Single-cell lines or simplified tissue models may not represent the diversity of human biology, e.g. in the genetic variability, organ interactions. Thank you. We have come to the end of today's video. Please don't forget to subscribe, don't forget to comment. Thank you.